The universe is full of surprises, moments that redefine our understanding of existence. It constantly challenges our perceptions and pushes the boundaries of what we believe is possible. Sometimes, a single announcement, a seemingly small event, can send ripples across our entire species, altering the course of history in ways we could never have predicted. These moments can be both exhilarating and terrifying. In November 2018, Chinese scientist He Jiankui, a relatively unknown figure at the time, stepped onto a stage in Hong Kong, ready to unveil something that would shake the foundations of modern science. With a mix of pride and perhaps a touch of apprehension, he declared the birth of the world's first genetically edited babies, a claim that would soon ignite a global firestorm. Lulu and Nana. Two innocent lives, unknowingly thrust into the center of a scientific and ethical debate that continues to rage to this day. This wasn't just another scientific paper, another incremental step forward in our understanding of the natural world. It was a fundamental shift in what it means to be human, a bold and controversial leap into uncharted territory that challenged our very definition of life itself. For millennia, we could only read the genetic code of life, deciphering its secrets like ancient scrolls, slowly unlocking the mysteries of heredity and disease. He Jiankui claimed he had edited it, not just read it, but rewritten a portion of the very blueprint of human existence. He altered the DNA of two human beings, making changes that would be passed down through generations, forever altering their genetic legacy. Before they were even born, intervening at the most fundamental level of development, raising profound questions about the ethics of genetic manipulation. The implications were staggering, a Pandora's box of possibilities and perils suddenly thrust open before our eyes, echoing through labs, sparking intense debates and frantic reevaluations of existing ethical guidelines. Governments and homes worldwide, forcing us to confront the profound societal implications of this groundbreaking yet deeply unsettling achievement. Had we crossed a sacred line, venturing into territory that should remain untouched, forever altering the natural order? Or taken the first step into a future free from genetic disease, a world where inherited illnesses are eradicated and human potential is unleashed like never before? The news landed like a meteorite, instantly dominating headlines and sparking global conversations. Both a technological achievement, a testament to the incredible power of human ingenuity and scientific advancement, and an ethical breach, a violation of long-held principles and a potential threat to the very fabric of our society. Scientists who pioneered CRISPR, the very technology that made this feat possible, were among the first to express horror and disbelief, recognizing the potential for misuse and the urgent need for responsible regulation. They had imagined this power would be used with caution, under strict ethical oversight, and with the utmost transparency, not in secret, behind closed doors, without proper consultation or peer review. Suddenly, the era of designer babies was no longer hypothetical, a distant possibility debated in academic circles. It had arrived, thrusting us headfirst into a future where the very essence of humanity could be subject to manipulation and design. This act catapulted humanity into a new chapter, a new era of genetic possibilities and ethical dilemmas. Written not in the stars, dictated by fate or chance, but in our DNA, subject to our own intervention and control. He Jiankui became a symbol of both ambition, the relentless pursuit of scientific progress, and the desire to push the boundaries of human knowledge, and reckless hubris, a cautionary tale of what happens when scientific ambition outpaces ethical considerations and responsible oversight. His work showed the power to rewrite the instructions, to alter the very code that governs our development, our health, and our future. That make us who we are, shaping our individual characteristics and defining our place in the grand tapestry of life. But it also served as a warning, a stark reminder of the potential consequences of unchecked scientific ambition and the critical importance of ethical boundaries. With great power comes great responsibility, a principle that must guide our every step as we navigate this new era of genetic possibilities. Our actions now could alter the future of our species forever, shaping the genetic destiny of generations to come and defining what it truly means to be human. To truly grasp the magnitude, the sheer audacity of He Jiankui's announcement, we must first understand CRISPR, what it is, how it works, and why it's so revolutionary. Inside every cell, the fundamental unit of life, lies DNA, a colossal instruction manual. the blueprint for everything that cell is and will become. It's billions of letters long, a complex code that dictates our traits, our predispositions, everything. For decades, scientists dreamed of editing this code, of rewriting the very essence of life. But the tools available were crude, imprecise, and often ineffective. It was like trying to perform surgery with a hammer. Then came CRISPR, a revolutionary technology, a game changer, adapted from the ingenious defense mechanisms of bacteria, where it serves as a vital defense against invading viruses, protecting them from genetic attacks. Scientists, with their relentless curiosity, turned this bacterial system into a molecular GPS, capable of pinpointing specific locations within the vast expanse of the genome and equipping it with a pair of microscopic scissors capable of making incredibly precise cuts. CRISPR can be programmed with remarkable ease to find a specific DNA sequence, no matter how small or hidden, and then, the Cas9 protein, acting as the molecular scissors, makes a precise cut at that exact location. This allows scientists to disable or rewrite genes, to correct errors or introduce new functions with unprecedented precision and control. CRISPR-Cas9 is cheap, making it accessible to labs around the world, effective, delivering results with remarkable consistency, and has fundamentally transformed biological research, accelerating discoveries at an astonishing rate. It's being used to develop targeted therapies for cancer, offering new hope in the fight against this devastating disease. Engineer resilient crops that can withstand harsh climates and resist pests, ensuring food security for a growing population, and unlock the deepest mysteries of life, revealing the intricate mechanisms that govern our existence. But this same power, this incredible potential, opens a Pandora's box of profound ethical dilemmas. If we can edit one person's genes to cure a disease or prevent a hereditary condition, what stops us from editing embryos, from making changes that will be passed down to future generations? Potentially changing the future of entire generations and perhaps the very course of human evolution? The line between therapy, aimed at curing disease, and enhancement, aimed at improving traits, is incredibly thin and easily blurred. The question is no longer simply if we can manipulate the human genome, because we clearly can, but if we should wield this power, and what the long-term consequences might be for humanity. He Jiankui didn't target a fatal genetic disorder such as cystic fibrosis or Huntington's disease, conditions that inflict immense suffering. He chose HIV, a virus that, while serious, is manageable with modern medicine. He was aiming to give children natural resistance to this virus, a preemptive strike against a potential threat. He recruited couples where the father was HIV positive, a population where the risk of transmission to the child was a significant concern. He then turned to in vitro fertilization, using IVF to create embryos outside the human body. At the single-cell stage, when the embryo was just a microscopic ball of cells, his team used CRISPR-Cas9, the revolutionary gene editing tool. They used it to target the CCR5 gene, a specific sequence of DNA, a doorway HIV uses to infect cells, latching onto it to gain entry. Some people naturally have a mutation, known as Delta 32, a deletion in the CCR5 gene, that blocks this doorway and makes them resistant, essentially immune to HIV infection. His plan was to mimic this mutation in embryos, to replicate nature's protective mechanism, hoping to grant lifelong immunity, shielding them from HIV for their entire lives. The risks were enormous. The potential for unintended consequences loomed large. Off-target effects, where CRISPR edits the wrong part of the genome, mosaicism, where some cells are edited and others are not, and unknown long-term health consequences. Despite these significant ethical and scientific concerns, the team proceeded, pushing forward despite the potential dangers. They continued editing embryos and implanting them into the mother's wombs. In 2018, twin girls, Lulu and Nana, were born, the first gene-edited babies in the world. Later, it was revealed that a third gene-edited child was born as well, adding another layer of complexity to the situation. He had crossed a line, venturing into ethically fraught territory, altering the human germline, the very essence of human heredity. Changes that could be passed to future generations, forever altering their genetic makeup. The recipe for a new kind of human, one potentially resistant to HIV, had been written and tested, with real-world consequences. The world was left to grapple with the implications, to ask, just because we can manipulate the human genome, should we? The experiment was not just a scientific endeavor, it was a profound ethical challenge. It was a leap into the unknown, with potentially irreversible consequences. The consequences would echo far beyond the lab, impacting society and future generations. Humanity had entered uncharted territory, a new era of genetic manipulation with uncertain outcomes. He Jiankui's announcement triggered a tidal wave of condemnation. Scientists worldwide called his experiment premature, unethical, and unnecessary. The medical justification was weak. Safe ways already exist to prevent HIV transmission in IVF. The edits didn't perfectly mimic the protective mutation. Their effects were unknown. He had created new mutations, rolling the dice with children's lives. He violated a near-global consensus germline editing should not be attempted in humans yet. The Chinese government launched an investigation, declaring he had violated regulations and ethics. The summit meant for careful debate was overshadowed by a reckless experiment. Lulu and Nana's story became a cautionary tale about technology outpacing ethics. The consequences for He Jiankui were swift and severe. After a government investigation, he was convicted of illegal medical practices and sentenced to 3 years in prison. He served his sentence and was released in 2022. The fate of Lulu, Nana, and the third child remains secret. Their health is an ongoing concern. He Jiankui did not disappear. He set up a new lab and re-entered public life. He acknowledged moving too quickly, but remained unapologetic about his vision. He now seeks to develop gene therapies for rare diseases, asking for a second chance. His return poses a dilemma: shun him or guide his ambition toward responsible research. The story is not over. A new chapter has begun. The world is still grappling with the questions he raised. Why did he do it? He Jiankui insists he wanted to help people. He frames his work as a pioneering medical procedure, not a reckless gamble. He believes germline gene editing is a frontier to be explored, not feared. In his view, preventing genetic disease before birth is a moral good. He sees himself as a pioneer, willing to bear the cost for future progress. Critics see hubris, he sees vision. He argues that history will vindicate him, as it did for IVF and vaccines. The debate is far from settled. He Jiankui's story reveals the dual nature of scientific progress, hope and hubris. Supporters argue we have a moral duty to pursue gene editing to end inherited suffering. With regulation and debate, they believe we can use this power for good. Many medical advances were once feared, then embraced. Critics warn that we don't understand the genome well enough to make heritable changes safely. Off-target effects and unknown mutations could have catastrophic consequences. There's a risk of creating a genetic underclass designer babies for the wealthy, inequality written into our DNA. The line between therapy and enhancement is slippery. Some call for a global moratorium, fearing a dystopian future. The stakes are nothing less than the future of our species. The debate is urgent and unresolved. The birth of Lulu, Nana, and Amy made human gene editing a reality. We've crossed a threshold. There's no going back. The technology exists, and its use is spreading. Will gene editing become routine or remain forbidden? The potential to end genetic disease is real, but so are the dangers. The human genome is complex. A single mistake could have lasting consequences. The risk of new diseases or social divides is real. Who decides what to edit and who gets access? These are questions of justice, equality, and what it means to be human. Our choices now will shape the future of humanity. We stand at a crossroads, holding the power to edit our own evolution. The story of He Jiankui is a lesson in ambition and the need for ethical reflection. We need a global conversation, one that includes everyone, not just scientists. International frameworks must balance preventing recklessness with allowing responsible research. The universe doesn't care what we choose. The responsibility is ours alone. Do we embrace this power or declare the human germline off limits? The story of Lulu and Nana is just the beginning. A wake-up call for our civilization. The tools to shape our destiny are in our hands.